Hello friends and welcome to the lecture on the topic entitled Molecular Methods of Assessing Microbial Phylogeny and Molecular Chronometer. Dear students, let's try to know about the major objectives of this lecture. Firstly, to get an insight about the molecular phylogenetics. Secondly, to understand different techniques used for assessing microbial phylogeny. And lastly, to get some knowledge about molecular chronometers. To begin with, let me give you a brief introduction. The biosphere is dominated by microorganisms and contains about 4 to 6 into 10 raised power 30 prokaryotic cells. This number represents at least 2 to 3 orders of magnitude, more than all of the plant and animal cells combined. Thus, microorganisms are a highly diverse group of organisms and constitute about 60% of the Earth's biomass. Owing to such enormous number, microorganisms are essential components of the Earth's biota and represent a large unexplored reservoir of genetic diversity. Understanding this unexplored genetic diversity is a high priority issue. Therefore, a plethora of biochemical and molecular methods have been applied to reveal the microbial community. Molecular phylogenetics is the branch of phylogeny that analyzes hereditary molecular differences, mainly in DNA sequences, to gain information on an organism's evolutionary relationships. The result of a molecular phylogenetic analysis is expressed in a phylogenetic tree. Molecular phylogenetics is one aspect of molecular systematics that also includes the use of molecular data in taxonomy. Early attempts at molecular systematics were also termed as chemotaxonomy and made use of proteins, enzymes, carbohydrate and other molecules that were separated and characterized using techniques such as chromatography. These have been replaced in recent times largely by DNA sequencing, which produces the exact sequences of nucleotides or bases in either DNA or RNA segments extracted using different techniques. Now, dear students, let's try to know in detail about different molecular methods of microbial community analysis. The vast majority of microbial communities in nature have not been cultured in the laboratory. Therefore, the primary source of information for these uncultured but viable organisms is their biomolecules, such as nucleic acids, lipids, and proteins. Culture-independent nucleic acid approaches include analysis of whole genomes or selected genes such as 16S and 18S rRNA, ribosomal RNA, for prokaryotes and eukaryotes respectively. Based on the comparative analysis of these rRNA signatures, cellular life has been classified into 3 primary domains, one eukaryotic, that is eukarya, and 2 prokaryotic, that is bacteria and archaea. Broadly, these techniques have been classified into 2 major categories depending on their capability of revealing the microbial diversity, structure and function. Firstly, partial community analysis approach and whole community analysis approach. Firstly, we will try to understand the partial community analysis approach. These strategies generally include polymerase chain reaction, that is PCR based methods where total DNA RNA extracted from a sample is used as a template for the characterization of microorganisms. PCR amplification of conserved genes such as 16S rRNA from a sample has been used extensively, for example, in microbial ecology, primarily because these genes are all ubiquitous, that is present in all prokaryotes. Secondly, are structurally and functionally conserved. And lastly, contain variable and highly conserved regions. Dear students, the partial community analysis approach includes following methods. Firstly, clone library method, genetic fingerprinting techniques, qPCR, that is real-time PCR, fish dot blot hybridization, microbial lipid analysis, DNA microarrays, microautoradiography and isotope array, DNA RNA stable isotope probing, and lastly card fish and Raman fish. Now dear students, let's get a detailed insight about these techniques. Firstly, the clone library method. The most widely used method to analyze PCR products amplified from a sample is to clone and then sequence the individual gene fragments. The obtained sequences are compared to the known sequences in a database such as GeneBank, Ribosomal Database Project, RDP, and Green Genes. Typically, cloned sequences are assigned to phylum, class, order, family, subfamily, or species at sequence similarity cutoff values of 80, 85, 90, 92, 94, or 97% respectively. Typical clone libraries of 16S rRNA genes contain fewer than 1000 sequences and therefore reveal only a small portion of the microbial diversity present in a sample. Despite its limitations, for example, labor intensive, time consuming and cost factor, clone libraries are still considered the gold standard for preliminary microbial diversity surveys. Genetic fingerprinting techniques. Genetic fingerprinting generates a profile of microbial communities based on direct analysis of PCR product amplified from environmental DNA. These techniques include DGGE or TTGE, that is denaturing or temperature gradient gel electrophoresis, single strand conformation polymorphism SSCP, random amplified polymorphic DNA RAPD, and DNA amplification fingerprinting DAF, amplified ribosomal DNA restriction analysis ARDRA, length heterogeneity PCR LHPCR, and ribosomal intergenic spacer analysis, terminal restriction fragment length polymorphism TRFLP, and produce a community fingerprint based on either sequence polymorphism or length polymorphism. Now let's discuss another partial community analysis method, that is DNA microarrays. DNA microarrays have been used primarily to provide a high throughput and comprehensive view of microbial communities in samples. The PCR products amplified from total environmental DNA is directly hybridized to known molecular probes which are attached on the microarrays. After the fluorescently labeled PCR, amplicons are hybridized to the probes. Positive signals are scored by the use of confocal laser scanning microscopy. The microarray technique allows samples to be rapidly evaluated with replication, which is a significant advantage in microbial community analysis. DNA microarrays used in microbial ecology could be classified into two major categories depending on the probes: 16S rRNA gene microarrays and functional gene microarrays. Now the quantitative PCR. Quantitative PCR, qPCR or real-time PCR has been used in microbial investigations to measure the abundance and expression of taxonomic and functional gene markers. Unlike traditional PCR, which relies on endpoint detection of amplified genes, qPCR uses either intercalcating fluorescent dyes such as SYBR, green or fluorescent probes TACMAN, to measure the accumulation of applicants in real time during each cycle of PCR. Software records the increase in applicant concentration during the early exponential phase of amplification, which enables the quantification of genes when they are proportional to the starting template concentration. When qPCR is coupled with a preceding reverse transcription, that is RT reaction, it can be used to quantify gene expression, RT-qPCR. qPCR is highly sensitive to starting template concentration and measures template abundance in a large dynamic range of around 6 orders of magnitude. Now, fluorescence in situ hybridization or FISH. Fluorescence in situ hybridization enables in situ phylogenetic identification and enumeration of individual microbial cells by whole cell hybridization with oligonucleotide probes. A large number of molecular probes targeting 16S rRNA genes have been reported at various taxonomic levels. The FISH probes are generally 18 to 30 nucleotides long and contain fluorescent dye at the 5C end that allows detection of probe bound to cellular rRNA by epifluorescence microscopy. In addition, the intensity of fluorescent signals is correlated to cellular rRNA contents and growth rates, which provide insight into the metabolic state of the cells. FISH can be combined with the flow cytometry for a high resolution automated analysis of mixed microbial populations. Low signal intensity, background fluorescence and target inaccessibility are commonly encountered problems in FISH analysis. It was all about the fluorescent in situ hybridization. Now let's know about microbial lipid analysis. Microbial community characterization by biomolecules other than nucleic acids such as lipids has been used without relying on culturing. Fatty acids are present in a relatively constant proportion of the cell biomass, and signature fatty acids exist in microbial cells that can differentiate major taxonomic groups within a community. The fatty acids are extracted by saponification followed by derivation to give the respective fame, which are then analyzed by gas chromatography. The emerging pattern is then compared to a reference fame database to identify the fatty acids and their corresponding microbial signatures by multivariate statistical analysis. Now, microautoradiography. Microautoradiography (MAR) relies on the fact that metabolically active cells utilizing radiolabeled substrate can be visualized by exposure to radiation-sensitive silver halide emulsions. The emulsion is placed on the top of cells that are mounted on a microscope slide. After exposure, excited silver ions precipitate as black grains of metallic silver inside or adjacent to the cells that can be observed by transmission electron microscopy. Commonly used radiolabeled substrates include glucose, acetate and amino acids, which provide a general view of the overall metabolic diversity. When MAR is used in combination with FISH, that is MAR-FISH, it allows simultaneous phylogenetic identification of active cells that consume the radioactive substrate. And lastly, the isotope array. Isotope arrays allow for functional and phylogenetic screening of active microbial communities in a high throughput fashion. The technique uses a combination of SIP, that is stable isotope probing, for monitoring the substrate uptake profiles and microarray technology for deciphering the taxonomic identities of active microbial communities. This was all about partial community approach. Now let's understand another important analysis method, the whole community analysis approach. Whole community analysis approach Sequence analysis of 16S rRNA genes is commonly used in most microbial, that is ecological surveys. However, being a highly conserved molecule, the 16S rRNA gene does not provide sufficient resolution at species and strain level. Whole genome molecular techniques offer a more comprehensive view of genetic diversity compared to PCR-based molecular approaches that target only a single or few genes. These techniques attempt to analyze all the genetic information present in total DNA extracted from an environmental sample or pure culture. These techniques are as follows: the DNA-DNA reassociation, G plus C fractionation, whole genome sequencing, metagenomics, metaproteomics, proteogenomics, metatranscriptomics. Now let's discuss them one by one. DNA-DNA hybridization kinetics. Whole genome DNA-DNA hybridization (DDH) offers true genome-wide comparison between organisms. A value of 70% DDH was proposed as a recommended standard for bacterial species delineation. Typically, bacterial species having 70% or greater genomic DNA similarities usually have greater than 97% 16S rRNA gene sequence identity. In DDH technique, total community DNA extracted from an environment sample is denatured and then incubated under conditions that allow them to hybridize or reassociate. The rate of DNA reassociation is correlated with the genome complexity, diversity present in the sample. Now, guanine plus cytosine content fractionization. Different prokaryotic groups differ in their guanine plus cytosine, that is G plus C content of DNA, and phylogenetically related bacterial groups only vary by 3 to 5% in their G plus C content. Thus, the fractionation of total community DNA can be achieved by density gradient centrifugation based on G plus C content. The technique generates a fractionated profile of the entire community DNA and indicates relative abundance of DNA, hence DEXA, as a function of G plus C content. The total community DNA is physically separated into highly purified fractions, each representing a different G plus C content that can be analyzed by additional molecular techniques such as DGGE or ARDRA to better assess total community diversity. Now, whole microbial genome sequencing. Exploring microbial systems through whole genome analysis is a comprehensive and integrated approach to understand microbial ecology and function. Whole microbial genome are sequenced using a shotgun cloning method that involves, firstly, extraction of DNA from pure cultures, secondly, random fragmentation of obtained genomic DNA into small fragments of approximately 2 kb, and thirdly, ligation and cloning of DNA fragments into plasmid vectors, and lastly, bidirectional sequencing of DNA fragments. Whole genome sequencing has provided unprecedented insights into microbial processes at the molecular level and has potential applications in individual and community ecology, bioenergy production, bioremediation, human and plant health, and various industries. Now, metagenomics. Metagenomics is the investigation of collective microbial genomes retrieved directly from environmental samples and does not rely on cultivation or prior knowledge of the microbial communities. Metagenomics is also known by other names such as environmental genomics or community genomics or microbial ecogenomics. In principle, Metagenomic techniques are based on the concept that the entire genetic composition of the environmental microbial communities could be sequenced and analyzed in the same way as sequencing a whole genome of a pure bacterial culture. Metagenomic investigations have been conducted in several environmentals such as soil, the phylosphere, the ocean, and acid mine drainage, and have provided access to phylogenetic and functional diversity of uncultured microorganisms. Post-genomic approaches. The recent application of DNA-based molecular techniques such as metagenomics have revealed new insights into the phylogenetic and functional diversity of microbial communities. However, in the post-genomic era, the limitations of DNA-based molecular approaches have been realized. For example, DNA-based techniques do not provide information on the gene expression as it occurs under in situ conditions. With the availability of comprehensive metagenomic databases, which also include genomic sequences from uncultured microorganisms, it is now possible to apply post-genomic approaches such as metaproteomics and metatranscriptomics to reveal the link between genetic potential and functionality in microbial communities. Metaproteomics, also commonly known as environmental proteomics, deals with the large-scale study of proteins expressed by microbial communities at a given point in the time. Compared to other cell molecules such as lipids and nucleic acids, protein biomarkers are more reliable and provide a clearer picture of metabolic functions than functional genes or even the corresponding mRNA transcripts of microbial communities. Now, proteogenomics. In metaproteomics, protein sequences could be identified with confidence only if they have significant homology to existing proteins in available databases. However, in most of the environmental proteomics surveys, proteins are only distantly related to known database sequences. Therefore, it appears that the majority of short protein sequences retrieved from metaproteomics will remain unidentified and cannot be assigned to the functional and phylogenetic features. However, these limitations have been overcome by combining the metaproteomic and metagenomic approaches together under the name of proteogenomics. In committee proteogenomics, total DNA and proteins are extracted from the sample, which allows linking of biological functions to phylogenetic identity with greater confidence. Metatranscriptomics. Metatranscriptomics or environmental transcriptomics allows monitoring of microbial gene expression profiles in natural environments by studying global transcription of genes by random sequencing of mRNA transcripts pooled from microbial communities at a particular time and place. Metatranscriptomics is particularly suitable for measuring changes in gene expression and their regulation with respect to changing environmental conditions. Now, dear students, let's learn about another section of this lecture, that is molecular chronometers. A phylogenetic marker is a highly conserved protein, that is ubiquitin, or nucleic acid, for example rRNA, whose rate of mutation is constant and which can therefore be used to construct phylogenetic trees. All sequences are not of equal value in determining phylogenetic relationships. To be a useful chronometer, a molecule has to meet certain specifications. Firstly, clock-like behavior, that is, changes in its sequence have to occur as randomly as possible. Secondly, range, that is, rates of change have to be commensurate with the spectrum of evolutionary distances being measured. And lastly, size, the molecule has to be large enough to provide an adequate amount of information and to be a smooth running chronometer. Now, rRNAs are the ultimate molecular chronometers. rRNAs are at present the most useful and most used of the molecular chronometers. They show a high degree of functional constancy which assures relatively good clock-like behavior. They occur in all organisms and differ positions in their sequences change at a very different rates, allowing most phylogenetic relationships, including the most distant, to be measured, which makes their range all-encompassing. This was all about these techniques. Hope you have understood them well. Thank you.